Okay, now I'm recording. Glad I remembered that. There we go.
Every membrane in the cell basically works in the same way. But most of the time in this class, I'm going to be focusing on the plasma membrane as the example. But if this was the mitochondrial membrane, that basically works the same way as the cell membrane. Or if this was a membrane within the endoplasmic reticulum, their basics are the same. So the membrane has to be doing several things at once. Besides just being a barrier. So it is a barrier, but it has to let outside information, right there, outside information about what the world outside the cell is like. That information has to be sent into the interior of the cell so the cell can use that info. The membrane has to allow import and export, and this is basically nutrients and waste. That's how to think about that. Nutrients, import, wastes, or secretions are going to be exported. Certain things in, certain things out. So last unit, when we talked about lysozyme, you've got lysozyme in your saliva, you've got lysozyme in your tears, somewhat in your nasal secretions. Lysozyme is a protein that gets made inside the cell. There it is right there. And the cells export it outside themselves so that it can protect from bacteria in that location. So that would be something that the cells have to make inside and send it outside. Export, we're going to talk about it in a different unit. That's unit 3. Import, we talk about some here and in unit 3. And then the plasma membrane has to be able to accommodate if the cell needs to move or if the cell needs to grow. It's got to expand or shift over if the cell needs to move or grow. This one I don't talk about as much in this class, but these first two you'll end up learning a lot about. Plasma membrane has to do many things simultaneously. It's definitely fluid mosaic. That's something you already know. It's fluid because it has motion. It's mosaic because it has protein. The main point of this slide is that there's a lot of protein. I feel like Gen Bio books show a sea of lipids and then a protein here and there. There's a lot of protein. And the proteins in the fluid mosaic are doing the jobs listed here. It's really the proteins that are doing the interesting things to receive info, import, export, allow movement. The lipids are more boring. They're there, but it's the proteins in the plasma membrane that are doing more interesting jobs. This is also usually something that students know before they come in, that the membrane is largely made out of these phospholipids. And they have a polar end and a non-polar end. Or they have a hydrophilic, water-loving end and a hydrophobic, water-shunning end, and that's still the case. Two things I want to point your attention to. The glycerol in here is like the building base for these. It's 3 carbon molecule, 2 of them are used to join these hydrocarbon tails. The 3rd carbon joins the phosphate head group. And then up there where you see choline, the choline is an example of a like a specialization on the phospholipid. Draw a circle around choline. Get this down so I can draw a circle around choline. That part is going to be changeable for different lipids. It's changeable for different phospholipids. Sometimes choline, sometimes some other chemical. This is also changeable. The amount of bend is also changeable for different phospholipids, so I might as well leave that there. When you have, I guess it's Gen Chem, and you learn about bond angles, you got your straight bond angles, and then you've got your angled bond angle. Every time you have a double bond somewhere in the chain, that's going to give you a kink in the chain. And I like kind of show it like, there's my kink in my leg, like that. Just keep my arms down, like that. Every time a phospholipid has a kink in one or both tails, that's going to give mobility extra mobility to the plasma membrane lipid. So the difference is if you've got 2 straight tail hydrocarbon chains and both are straight versus this, these pack tight. Straight chains pack tight. Kinked chains give more mobility because they don't pack tight together. So if we wanted to get as many people as possible in a room, number one, we wouldn't do that right now, but if we did, we could get the most people in and have them the most frozen if everybody was standing totally straight and they don't have bags and they don't have like crutches or backpacks or umbrellas with them hanging off. Totally straight is totally more packable. So this type of stuff, straight versus kinked, has a lot to do with lipid movement in the membrane. And this is the best picture for me to show that at the moment. This is something we already talked about earlier in the semester. It's all the same. I'm going to skip past that. But this fact about water caging being unfavorable for hydrophobics, that fact forces phospholipids into a bilayer. That's why the bilayer forms. So having all the hydrophobic tail regions together shields them from water. And you've got the hydrophilic groups on one side, it's kind of like a sandwich. So this behavior of the lipids, it's always going to form two compartments. You've got the outside watery compartment, the inside watery compartment. So this could be the outside of the cell and this be the cytoplasm. Or if this is the membrane of an organelle, this could be the cytoplasm and this could be the inside of the organelle. So something really cool about phospholipids is if you put a few of them into any aqueous solution, they will spontaneously form tail to tail, heads sticking out. They will spontaneously arrange that way, and they will form micelles or vesicles, which are like this. So we could call these micelles, or we could call these vesicles. And it's a sphere of phospholipids with an inside compartment, outside compartment, and that's spontaneous. There's a lot of cosmetic application now using what's called micellar water. So if you've ever bought that, you can use it to clean off your sweat, your makeup, or whatever. That's just lipids in droplets that are basically vesicles. And when you put it on your face, this end will be attracted to grease and this end attracted to water, and it's like a mild detergent action to clean off whatever's on your face. That's so useful because it used to be that a lot of people have heard of this. Vesicles are more like, what is that? They're the same thing, basically. So the chemical properties and the chemical behavior of those phospholipids give so many properties. So this is the tendency. This is the tendency for a collection of phospholipids to do, is to form either a small vesicle or a micelle or a whole plasma membrane around the cell. And if the plasma membrane rips or gets disrupted in some way, it will automatically try to reform this sphere. It will automatically try to self-seal without the cell having to do work to do that. That is so cool. So if you're a cell and this should be your normal configuration, and you've got a little rip in your plasma membrane, that can be self-healing if it's not too damaging. You can fix your own rip if it's not too gigantic where you lose your homeostasis. It's really helpful for cells. So that's kind of big picture stuff about membranes. The next thing I want to get into is how movement works in membranes. The membranes are moving and the components of the membranes are moving a lot. And I think you had a quiz question about this, like what are something about the movement. Which ways can it move? I think it's which way can it not move? No? Oh, okay, good. That reminds me, you've got some quizzes coming up that are fill in the blank or type in your answer, because the question for the quiz used to be, name 2 or 3 ways that membrane lipids could move. So I've been trying to convert them to multiple choice, but some of them it's just, I haven't. So when you come to quizzes that you have to type in an answer, I have to grade those myself. And it's going to look like you get it wrong. So don't freak out about that. There's some of those coming up in this unit. So just remember, this goes for everybody in the class. If you have a quiz and you have to type in the answer or fill in the blank, it's going to auto grade as being wrong at first until I go in and change it. So just be aware. It doesn't mean that you got it wrong. I just have to grade those myself. So when I'm thinking about plasma membranes and how they behave, I think that they're like people, because they kind of got a head and they've got legs like so. And the first thing to write on here is that we call each one of these layers of phospholipids a leaflet. And that's maybe a new terminology. So if this is the cell membrane, and the outside of the cell is up there, if that's the case, we would call this the outer leaflet. And this would be the inner leaflet. It's all one plasma membrane, but each half of it can have some different properties. And if you're trying to talk about half of the plasma membrane, it's called leaflet. So most of the motion within the plasma membrane occurs within the same leaflet. So the lipids can kind of go past each other. So this would be people jostling in a crowd. You could be packed together but still moving individually through the crowd. So that's the lateral. Individual ones can spin around. Rotation. Their legs can flex side to side somewhat. That's the part that depends how many double bonds, how much flexion do you have. But the difficult motion is to move from one side, one leaflet to the other. That's a more rare motion. This one is controlled by that enzyme called flipase. Best enzyme name, flipase. Why is that movement difficult when the other ones are not?
The one's going to take more energy.
It takes more energy. It does take more energy.
And you got to get the hydrophilic head all the way across the membrane.
Yes, so you have to get a hydrophilic head group through the hydrophobicness of this interior. That's unlikely. That's energetically more expensive, and that's why there has to be an enzyme to do that. These other movements, the diffusion and the flexion and the rotation, they're more naturally occurring movements. So when all of this together means that the plasma membrane lipids are in constant motion. And in that video that we watched at the beginning of the year, that's one of my favorite things to look about it, because they show the plasma membrane as almost, it's like a liquid sea of motion, and that's the realistic state of it. You got these membrane lipids moving in different ways, and you have lots of proteins in this, not shown here, but you have lots of proteins in the fluid mosaic moving laterally as well. So when you think about membranes in this class, think about them as being in motion unless something is anchoring the protein down. They are in motion. Assume that motion is the default. One thing that can stop the motion or slow down motion in a membrane is cholesterol. There we go. Plasma membranes need cholesterol in them. It's a normal part of the membranes. This fact makes cholesterol essential. This is like the therefore thing. What's that called in math? Okay. It's been a long time, but I use this a lot. Cholesterol is essential for cells, for human cells. And we do need a certain amount of it in our diet. Or we need fat in our diet so that we can build cholesterol. Our liver does that. And you need building block amounts of it. Every membrane in your body has some amount of cholesterol in it. This is the base. This part of cholesterol right here is a really important chemical building block for a lot of things in our body. When cholesterol is in a membrane, it's got this hydrocarbon tail part that helps it stay in the membrane. And this shape is not going to flex. That's kind of a rigid shape. This is flexible, this is not. But notice, the overall shape of this is a space filler that fits really well between a kinked hydrocarbon chain and a straight. It's kind of a beneficial space-filling molecule that can fill that gap. And when the more cholesterol you have, the less fluid the membrane movement. Is that up there? Kind of. More cholesterol in the membrane gives less fluidity or less motion to that membrane. Look at what picture I've got next before I add something here. So you can have regions of the plasma membrane that have more cholesterol than the surrounding regions. And I'm going to draw a lame picture of this. So pretend that this is all plasma membrane that I'm drawing in this tacky way. And this region has more cholesterol These regions on either side have less cholesterol. And those regions of the plasma membrane are kind of going to act differently. This region is going to kind of stay together more solidly. There's less movement within that region, and the cholesterol is causing better packing, better stability in this region. These regions are more fluid because they have less cholesterol. So this kind of arrangement in the cell is called a lipid raft. This thing right here, that's called a lipid raft. And lipid raft can organize certain proteins together that need to stay together. And they kind of float around like an island. This lipid raft will float around like an island in the bigger membrane. These are useful for keeping, let's say, functional sets of proteins together. Kind of like how teams of EMTs, or you got your ambulance crew. They need to stay together. They're supposed to work together, so they ride around everywhere in the ambulance together. And when they get to the place, they do their job together. That's kind of like a lipid raft. So lipid rafts kind of float through the bigger membrane, carrying functional sets of proteins that are supposed to work together. And there's a really nice animation of that in that starting video. I may play that video after the break today. Lipid rafts are kind of new, and they're not in our book, but they're turning out to be pretty important to cell biology. This was also one of your quiz questions, and the answer was each side of the membrane has different chemical properties or something like that. So you can see in here, we've got this leaflet, this leaflet. They're showing cholesterol in the gray, and they're showing different colors of the head groups. And some of the head groups have been replaced by disaccharides, sugars. Depending on the cell and what it does and what its jobs are, a lot of times the outer surface of a cell membrane that faces the outside world is going to have a lot more sugars or a lot more carbohydrates on the outside. And these sugars, these carbohydrates, have two big functions. One of them is they help hydrate the cell surface. They're big and they're hydrophilic, and they keep water close to the surface of the cell. So they have a hydration function. And your sugar, your carbohydrate surface can help serve as cell identifiers. The arrangement of sugars on the outside of the cell can help your body differentiate that it's a human cell, it's not a human cell. That's a shape. It's a shape, 3D shape surface of its own, and it's like an identifier. Those are going to be on the outside surface of the cell, the carbohydrate stuff. So in this picture, this would be exterior of the cell. The interior of the cell doesn't need carbohydrate, but it might need different chemical head groups on the lipids. These would be different chemical groups than the ones here. And that's the basic thing you need to know for this class.
Okay, 2 more slides and then our break.
This slide is talking about what lipase does, which we already talked about. It can move lipids from one side, one leaflet to the other. And the main place where lipase works in a cell is in the endoplasmic reticulum. The endoplasmic reticulum is really important for actually constructing lipids, constructing membrane components. Cells have to always be making more membrane, just as a matter of fact, to replace worn out membrane. And the place where cells make new membrane is endoplasmic reticulum. So endoplasmic reticulum constructs membrane lipids, and then it sends membrane lipids out to the place where they're needed. So I want to write that in the slide. ER builds lipids, membrane lipids. And it sends them to locations where they're needed. So maybe the cell needs more membrane lipids out of the cell membrane. Maybe it needs more membrane lipids for the mitochondria. Those are different locations, but the ER would be building all of the lipids. And when it sends the lipids, it's sending them in vesicles. Individual lipids don't flow through the cell. They form vesicles and they migrate through the cell. But to do this, it's going to be a gorgeous picture right here. Here's the ER, and it's got all these connections and lovely nature. The vesicle has to form out of this membrane. There are not membrane lipids just kind of floating around in the cell. The vesicle has to form out of this membrane. And to do that, it's helpful if you make the outer leaflet have more lipids than the inner leaflet. This is going to help start the bulge to form that vesicle. So that's where you need lipase, either to help form the bulge, or as the bulge is getting too big to flatten it back out. So if lipase is acting in the ER, it can help membranes start to bulge, or it can help membranes stay equal on both halves. So it's kind of like the default place to put new lipids is on the cytosolic phase. But if you need to balance out the curvature, lipase does that.
this is what I was kind of trying to draw. This picture is very important in the colors of these two. There's a light orange color and there's a darker orange color right here. And they have a really important meaning for this concept. Every plasma membrane has those leaflets, the inner and the outer. And every leaflet has a particular chemical nature, like we saw here. They're chemically different inside from outside. Go back here for a second. And any proteins that are in here also have an orientation. So if there's supposed to be a protein in this membrane that is a transporter, that's moving things from outside to inside, it has to be oriented the correct way. The protein could be the right way or it could be upside down, for example. So it's super important to maintain the orientation of this membrane. So when membranes are being built in the ER, they have to be built with the correct orientation. And that's what these orange and darker orange colors are trying to show here. So the inside of the ER is always going to be the side that is representing inside a compartment or outside the cell. So let me explain what I mean by that. What's inside here on this leaflet that they're showing in the light orange, that's always going to be equivalent to the inside organelle leaflet or extracellular. That lighter orange. And here's how to kind of see that. So if a vesicle is forming, pinching off, once it forms, it's got an inside and an outside. And when this goes and fuses with a new membrane, what's inside goes into that compartment or goes outside the cell. So this would be extracellular over here. This leaflet that's on the outside of the ER is going to be the one that's cytoplasmic facing. And that never changes. No matter where the vesicle is going, the outside leaflet on that vesicle is going to be cytoplasmic, always. So it was cytoplasmic facing in the ER. It's cytoplasmic facing on the vesicle, and it's cytoplasmic facing after vesicle fusion. So here's something else that makes this easier to see. Let's draw in some proteins. Membrane proteins. On each side of this leaflet. They're totally fake shaped. Just wanted to be different looking. So if the ER is building membranes this way, and also ER is also building in membrane proteins at the same time. We talk about that in a separate unit. But the ER is constructing membranes and proteins that go in it. Okay, so what I want you to do is draw these proteins in on the vesicle and on the fusion picture. Draw them in for yourself. That's, to me, is the best way that I can get my mind around this spatial orientation thing.
If their orientation is conserved, well, flip base, like change that on the previous slide.
Say that, the first part of that again.
So you said that the protein orientation on the membrane is like set, right? So it doesn't change.
Yeah.
So when the flip base kind of like reorients it, won't that change the protein?
Flip base only moves the individual lipids. So it moves individual lipids one at a time from one leaflet to the other. And the protein would stay stuck even if individual lipids around it are moving. But that's a good question. So flip base can't change the protein orientation. It can just move individual lipids between leaflets. Yeah. You think you've got this? Do you have the asterisk proteins over here facing the cytoplasm? Good. And then the blue blob proteins are facing extracellular. This slide is going to be really important when we come to the beginning of Unit 3, this particular slide. So let's have our break right now for 5 minutes, and we'll then resume. The copy shortcut on a Mac and a PC are not the same. So students that are on Teams, at the end of class today, I'm going to show this video again, and it would be better for you all to just watch it separately on your computer than try to watch it through the OWL. It will go a lot better that way. So end of class, I'll probably stop a little bit early. Go ahead and leave Teams and then watch that video. It's in the course resources module for cell biology. I put it at the bottom right here. Or I can share my screen real quick. Okay, so in cell biology in course resources module, there's the video link right there.
I got it, thank you.
And when you're watching it, just be looking for all of the membrane stuff that were included for today. It'll kind of pop out at you after this lecture. Okay, let me erase. Okay, so this second part of this lecture is talking more about the proteins in the membrane than the lipids. So we won't really talk about membrane lipids specifically anymore this semester. We'll spend more time talking about all the different proteins that make up the mosaic and that provide the jobs of the plasma membrane. So remember, you have to receive information. So this is an example of the cells getting information about what's outside. You have to move substances back and forth using transporters. And you have to have structural integrity. These have to do with cell signaling, these classes. So you could write on this side, the receptors and the enzymes have to do with cell signaling and information transfer. These have to do with movement of nutrients, wastes, secretions, the transporter proteins. And the anchor set, or the structural set of proteins, that's going to be more involved with cell division, movement, staying put. The cell needs to stay put. That's kind of their big categories. So this is going to be this unit 3, unit 4, somewhat. But the proteins are the more exciting part of the membrane. There is so much protein in a membrane. The membrane is very crowded with proteins, no matter if it's cell membrane or organelle membrane. And any protein that is with the membrane in some way, it differs in how much is it with the membrane. How stuck into the membrane is it? And these categories are really useful to know. So I'm going to try to write underneath here. If the backbone crosses all the way through the membrane, or let's say the backbone crosses both leaflets, that's called transmembrane protein. It's going all the way across. So when you see here, here's the alpha helix that goes all the way across both leaflets. This protein has 3 crossing alpha helices. This has beta sheets going all the way through multiple times. Those are transmembrane. If only one leaflet is being crossed through, let's say, that's called monolayer associated. I never heard of that as an undergraduate. It's kind of a newly discovered category. Monolayer associated. These two together, so monolayer associated and transmembrane, they fall into a bigger category called integral membrane proteins. Integral membrane proteins, because the backbone is with the membrane and they're really stuck in there. They are firmly associated with this membrane. There are other proteins that are kind of with the membrane, but they are not integral. They have some other association. So some proteins are covalently linked to a lipid, and the lipid is in the membrane. This zigzag red line, that's a membrane lipid. And this is representing there's a covalent attachment of the whole protein to one or more membrane lipids that go into the membrane. So these proteins are with the membrane, but they are not firmly wedged into that membrane. They would be kind of easy to rip off experimentally. Those are lipid linked. These are, in a way, covalently linked to the membrane. These are only held at the membrane by the 4 forces. So protein attached. I learned this category as called membrane associated. Actually, both of these categories, the more old school term is that they're membrane associated proteins. These are technically covalently held with the membrane. These are non-covalently associated by those 4 forces.
Both of those groups?
Only the protein attached.
So which?
Sorry, these are covalently associated with a lipid. These are attached to, these are associated with another protein that's really in the membrane. But this association between the gray and the green is non-covalent. It's 4 forces binding. Both of these classes together are membrane associated. So this group over here are integral membrane proteins, and this group over here are membrane associated. Kind of like, if you compare it to professors, like your full-time professors are king integral king teachers, and your adjuncts are like king associated teachers. They're not as permanently held in general. this group, it's a lot easier to purify the proteins away from the membrane. Because they're not that firmly wedged into the membrane. This group. This group is hard to separate or hard to purify the protein away from the membrane. One more thing. I always forget to, this is the best place to put this. Proteins that aren't associated with any membrane, if there is no membrane relationship for the protein, those we call soluble. Those are the soluble proteins. Soluble proteins float around, and they're not really joined up with membranes for the most part. They just float around. That's soluble. So every protein on here is not so much considered soluble. These could be soluble sometimes, but anything that's covalently with the membrane or it's lodged into a leaflet is not soluble. It's with the membrane semi-permanently.
So lipid link is covalently bonded.
To a lipid that's stuck in the membrane. Yeah. Let me show a picture that could also explain lipid linked. So if we look back at this leaflets picture, if these sugars were like a protein instead out there, that would be lipid linked. So what's holding it in is the position of one or two lipids in the leaflet. And that's a pretty solid attachment, but it's not as good of attachment as if the whole protein is wedged into both or one leaflet.
In that picture, are the carbs taking the place of the hydrogen?
Are the carbs taking the place? There could be some where it's two sugars exactly like that. There could be some where there's a head and then a sugar. So it's kind of oversimplified. And the types of possible phospholipids, there's really a lot of types. And people that study that are more chemists. They're kind of between chemists and biologists because it can be so diverse. They're organic. Those chemicals are organic. Yay. So if you're a protein that you're in one of these categories here, you are membrane, transmembrane, or integral membrane, your backbone is participating in the leaflet. So this would be one example of that. This can also be called membrane spanning. Is that on that previous slide? No. Okay, add on to this. Transmembrane is also membrane spanning. Membrane spanning, S-P-A-N-N-I-N-G. There's so many names for these, and I used to spend 30 seconds on this slide, and then I realized they really need a foundation in all the different names. Membrane spanning. So if you are a transmembrane or monolayer associated membrane spanning protein, you've got to have a hydrophobic region. because you are crossing through, your backbone is crossing through the interior of the plasma membrane, which is hydrophobic. So if you're going to stick there, you also have to be hydrophobic in that region, in this region. So this alpha helix, it continues. There's more of the protein here. There's more of the protein up off the screen. But this region right here, all of the R groups would have to be hydrophobic for this to work. And that's how they're constructed. So if you're looking at a protein sequence, like the primary sequence, and you see that there's a big stretch of hydrophobic amino acid, it might be a membrane spanning region. Alpha helices are a common way that proteins thread through a membrane. If you are a protein that needs to form a passageway for things to cross from out to in or in to out, there's a few different structures that can accomplish that. So one way to accomplish that is that you form a pore, aqueous pore, that is a watery connection between outside and inside. So the terminology of having a pore, it's like a tunnel with water inside. One way to make such a pore is to have multiple alpha helices in the protein structure crossing through the membrane, and these alpha helices would be mostly hydrophobic. So in this picture, the green parts are hydrophobic. The green parts are hydrophobic because those are the parts that are touching the interior of the plasma membrane. And one thing I don't love about this picture is that they're showing like hydrophobicness coming up out of the plasma membrane. These ought to be level with each other. Or these ought to be red if they're poking up out into the extracellular space. Or if these regions are poking down into the cytoplasm, they ought to be red because that's an aqueous, that's a watery environment. But the inside of this pore has to be hydrophilic amino acids because there's going to be water going through the pore. So this would kind of form a pattern like hydrophilic, hydrophobic, 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 hydrophilic. Like every fourth residue or something would be hydrophilic and the rest hydrophobic in this pore forming region.
What keeps it from slipping?
What keeps it from slipping? It's the hydrophobic associations.
Isn't that the same like the,
Say that again.
so when you have like hydrophobic green parts in
Uh-huh.
the previous slide,
Yeah,
it spans the whole like alpha helix, right?
this region.
So
Yeah.
what keeps it like stationary in the middle?
Yeah. So the reason that this doesn't slip up or down is the hydrophobic to hydrophobic interactions are entropically more favorable than if this were to slip down and these touch water. That's kind of an unsatisfying answer, but the hydrophobic interactions keep this in there, and it's actually really strong, really strong. Trying to get this protein out of the membrane is tricky, and it requires chemical help. It's really stuck in there. So it doesn't really shift up and down, but it could shift laterally. This could move laterally, but it's not going to move up or down within the membrane because those hydrophobic interactions are really strong. Good question. So if a cell needs a really big pore through a membrane, a really big hole, it tends to use beta sheet structure to form a big hole or channel. And in the same way, each side of the beta sheet is going to have a different nature. The side that interacts with the membrane would be hydrophobic, so that's the dark green on this is hydrophobic. And the side that interacts with the water coming through the channel is hydrophilic. And these are called beta barrel. I just like that. And I heard of it, it made me weirdly happy, like beta barrel, that's a great name. Name describes what it is. Most of the time, if you've got a big hole through a membrane, there are ways to shut it from one side. Otherwise, the cell can control what's in and out. So, big hole, you would have some kind of movable part on one side or the other to close it in some ways. Okay. So if you want to study membrane proteins, it's a pain. Excuse me, it's a pain because they're so wedged in the membrane. If you want to study them on their own, you have to get them out of the membrane on their own, and we're actually doing this today in lab. We're going to be studying proteins, and when you make a protein extract, a lot of times you'll use a detergent, some kind of detergent, and these are some examples. So this is the one what we're using today. Sodium dodecyl sulfate. What's dodecyl, like 12? 20? 12. Sodium dodecyl sulfate. And it's got hydrophilic head, hydrophobic tail. Another example is Triton X. We actually were using Triton X last week in our ELISA at a very low concentration. So these detergents can get in amongst the membrane lipids and break them up. They can get in there and break them up and help the protein get away from the plasma membrane lipids. They're breaking up the fluid mosaic, and the detergent can coat that hydrophobic region of the protein. So this would be the hydrophobic dark green region. So that hydrophobic region is shielded by those detergent molecules. And by using the detergent, the protein becomes soluble. It's away from the membrane and it can dissolve now, instead of being stuck in a fatty membrane. It's hard to see, but it's easier to see up there. There's a dark green region in that protein that represents the hydrophobic. And in this one, they've got it correct that the hydrophobic part lines up with the hydrophobic part of the membrane. They've got the levels right. So getting this protein out of the membrane, it's really stuck in there. You've got to use chemical help. And today when we're doing our protein extraction, there's going to be detergent in that buffer that's going to help membrane proteins get soluble so then we can load it in a gel. And remember, the one for today is sodium dodecyl sulfate, SDS. SDS is the detergent today for lab. This slide is in here just to show that a single transmembrane protein can do an amazing number of things at the same time. So we see here that this is integral. This is an integral membrane protein because its backbone is passing through the plasma membrane 7 times. 7 alpha helices pass through. And this protein is doing 3 things at once. It's absorbing light using this retinal coenzyme. And it's using light energy to directly pump protons out of the cell. So it's absorbing light. With the help of a coenzyme, retinal. It's kind of a derivative of vitamin A. And then it's using that energy directly to pump protons. So it's pumping protons and it's forming a proton gradient outside the cell. This word is forming. This is called bacteriorhodopsin. Bacteriorhodopsin, it's a bacterial protein. And when I went to graduate school, there were people in one of the labs that I could have rotated in, and they studied things like this. And I didn't know what this was. I'd never heard of it. I thought, man, that sounds kind of boring. I didn't understand what it was until way later, but this is amazing. This is the primitive ancestor photosynthesis protein, bacteriorhodopsin. And it is doing photosynthesis processes all in one protein. Capture light, pump proteins, and all you need with this is an ATP synthase, and you turn light into ATP. It's amazing. Had no idea what they were talking about. Those of you who are in plant, did you talk about this in plant? Of course you did. Han understood what it meant, and he rotated in that lab. The other reason I didn't rotate in that lab is that everybody in there was a jerk. Did he tell you about the guy with the trash can? Yeah, so there was a guy that studied this, and he had a science paper or a nature paper. That puffs your head up anyway, but he's big time researcher at Texas A&M right now, still studying the same stuff. And he was Chinese, and there was a Japanese guy that came to work in this lab because they were world renowned at this stuff. And the Japanese guy threw his Subway wrapper in the Chinese guy's trash, and he's like, "Don't use my trash." That was like the attitude of that whole lab. Amazing stuff. One complicated membrane protein can do a lot of things at once. It's just beautiful. So one of the things I want to do at King is to teach you all a little bit beyond just the stuff that you think you need to know. I'm going to skip this slide and go to this one. So we're skipping over this structural protein thing on this picture. We'll come back to that later in the semester. I want to talk about this concept. So if a protein doesn't have anything to hold it in a certain part of the membrane, it will drift. It will drift, and it's not the proteins moving, it's the lipids moving past it. Like if this room were full of people and I just stood still and all the other people were jostling around, I might find myself over by the door when I didn't try to go there. So if proteins don't have a way to make them stay put, they will drift. And these are some different ways that make proteins stay put, or make them stay in a certain spot on the membrane. All of these three here are working by non-covalent four forces interactions. So you can have inside anchoring spots. So in all of these, it's the green protein that we're looking at as the example. What's making that green protein stay put? So it could be that there are proteins inside the cell that are associating with that, and they're holding it in place. It could be proteins on the outside of the cell that are interacting and holding those green ones in place and not moving laterally. This is still outside of the cell, but it's two integral membrane proteins. They're actually the same, and they're holding each other together. All of those would keep this green protein from moving away, from drifting. The other way that you can keep proteins from drifting from place to place in the cell is to have tight junctions. And your book uses this black bar to show the concept of a tight junction. Tight junctions are integral membrane proteins, and their purpose is for being a diffusion, let's say a lateral movement barrier. They are a lateral movement barrier. And if this were a 3D cell, you would have a ring of tight junctions all the way around. So, number one, they are a lateral movement barrier. So that means that green proteins here can move in this area, but they can't go past. They could drift around anywhere up in here, but they will not go past this ring of tight junction. Yeah.
If the tight junction is an integral membrane protein, what's keeping that in place?
What's keeping that in place? They are anchored to the cytoskeleton. And that's such a good question. They're anchored to the cytoskeleton, which I haven't mentioned at all yet, but that's kind of like, I shouldn't have skipped over this. So proteins can link into the cytoskeleton, which is like the permanent, semi-permanent framework holding up the cell membrane. But tight junctions stay put where they are because they're associated with the cytoskeleton. So this is, you can think of tight junctions as a wall. They keep green up here. Oh no. Well, it'd be quicker to just erase the arrow. They keep green up here. So green proteins have to stay there. Blue proteins can go anywhere around here as long as they don't pass the tight junction. So it's segregating proteins. The other thing the tight junction is doing is that these prevent diffusion of substances between tissue compartments. So this tight junction is connecting to a neighboring cell. And if we have glucose is the example that we're gonna see. If we've got glucose up here, a bunch of glucose up here in this space, the glucose can go that far, but not past the tight junction. So we're gonna see this arrangement where this is gut lumen, not lemon, gut lumen. And down here could be this cell layer is part of the intestine. This is an intestinal cell. And then down here is your digestive blood supply. I'm going to set this up for next time. This will be where we stop today. We'll watch the video at the beginning of next class. Next time when we talk about transport, this is one of the major awesome arrangements of cell transport to move glucose always in one direction from your gut lumen into your blood supply. And the arrangement of these transmembrane proteins are really important for that. Don't forget this afternoon, come to lab, look for us in the classroom for pre-lab. Bring your ELISA work. That's a Canvas assignment, right?
Yeah.
Okay, yeah.
There was one question, who was the last one? Did you guys have the same problem that was trying to have you upload them?
Oh, yeah, I made like a Word document.
Like, there was no text box, it was,
I just typed it into
wrote
a
it
Word
down.
document.
I just worded it.
Oh, did that make a question like with no place to answer
yeah.
it? Okay.
It was like, who tested positive, and then we expect them to have symptoms.
Yeah, and there's nowhere to type.
Yeah, instead of like typing
Yeah.
an answer, it was like.
Let me fix that immediately.
Well,
I mean,
we already turned in something.
Yeah.
That's
It
fine.
lets you upload a picture, so.
Oh, it lets you up. I'm sorry.
It let me upload a Word document, I just typed it into a separate Word document.
I did not intend for you to have to upload a picture or a Word document for that, but.
Okay, so I had a problem with mine.
Mhm.
I accidentally deleted my picture.
Uh-huh. Of your
Test.
tray.
So I have everything else that I need. I just don't have that picture. So will that just like count off for the one question, or will it mess